Welcome, everybody, to another Pathfinder program. My name is Tommy Soares. I'm the Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And today, I have the pleasure of welcoming Dr. Heinz Reiske. He is the Global Director of Laboratory Operations, Genomics for Labcorp Drug Development. Thank you, Dr. Reiske, for joining us today, and thank you for taking time to tell us a little bit about yourself. We're eager to learn not only what you do today, but what was the journey that got you there. So thanks again for sharing and opening up to us today.
Sure, Tommy, happy to be here.
Well, Heinz, you know, the one thing that I would really like to know is, what is the path that got you to be this global director? And I'm so curious about what you actually do in this position. But, you know, if you could take us back maybe to what's your background, what's your training, and, you know, what got you in this particular career path? And let's start there and we can go
Sure.
from there.
Yeah, absolutely. That's so when I got started, I guess I'll go back to my postdoc.
Okay.
So when I came out of grad school, I went to Cornell in Ithaca, New York for my PhD. And the topic of my thesis was integrin-mediated signal transduction. And so when I went to the NIH for my postdoc, it was also in kind of a similar area, but studying a transcription factor that was important in cancer. And that kind of led me down the path to get into genomics testing. So when I was at the NIH, it was like 2000, real-time PCR was just kind of coming into its own. Like, people were still doing southern blots to confirm array data. And so I learned real-time PCR and then started to get exposure to all these other things in like bioinformatics tools. And I fell in love with that. And so from there, I got into biotech companies in the DC area. And that's where I got into really doing contract work. So among the first companies I was working for was kind of a small CRO. And then after leaving there, we relocated to the Boston area. And I got into development of molecular diagnostic tests, or what would be known as IVDs, or in vitro diagnostics. And that kind of set me to the path of clinical diagnostics, and then you know, really learning all the regulatory landscape, the technology. And the last company I worked for in Boston, it was a startup, kind of ran out of money. So I always wanted to know what it's like to be in a CAP/CLIA environment, and saw a job with what used to be called Covance and is now Labcorp Drug Development. And the rest is history, as they say. So here I am as a leader in genomics for Labcorp Drug Development. So it's what it really means is you manage people and the operations. So that's kind of a whirlwind tour of the last 20 years or so.
Wow. So from Ithaca, New York to Washington, D.C. to Boston to Indiana. You're in Indianapolis, is that correct?
Correct, yes.
Yeah, awesome. And so, Heinz, so take us back a little bit to that thesis. What was it about that thesis that you said, hey, you know, not only am I qualified, but I think I'm going to go try out this thing in DC. Like, I think I just want to go do this. What was that time in your life? And take us back. Well, like, what were you thinking?
Yeah, sure. So I kind of knew that I wanted to go into industry, even at the start of grad school. Like, you know, I knew that the tenure track route wasn't probably what I wanted to do. And so when I was looking around for postdocs back in those days, really, you had to kind of do a postdoc even if you wanted to get into industry. I thought the NIH was kind of a neat place. You had exposure to patients. So I actually had the project where I was able to go into the OR and work with the clinicians. Like they would take a tumor sample and hand it to me. So you really couldn't do that anywhere else. It was a kind of a different pace than if you had gone to like another university and another academic department. So I kind of thought the NIH would be a good, kind of bridge to, something which in those days was referred to as an alternative career, and now is more like the rule rather than the exception, I think. So that's what kind of led me down that path. And even studying signal transduction in like the late 90s, there was an obvious translation of that in, cancer cell biology, and knowing that some of these proteins that we were studying in cultured cell lines were going to be drug targets someday. And certainly in the work that I do now, I kind of chuckle occasionally how I've come full circle looking at some of the targets of these drugs. I was like, oh, I remember doing a Western blot on that particular protein and which antibodies worked and which ones didn't.
That's awesome. That's awesome. And I think, it's so true, the evolution of biology, biological research has just accelerated it so quickly. Was it hard applying for this NIH postdoc? Was that a difficult process? Do you recall?
Not really. It was, at that time, there seemed to be quite an appetite from various places to have, expand postdocs. It's a fairly easy transition to start at the lab. The people there were great, very nice, very passionate about what they did. And they love to take people on tours of the building and what the labs did. It was really a special place. And for anybody watching, I would highly recommend NIH and Bethesda as a destination if you're considering postdocing, for sure.
But that's awesome to hear. And I think you're probably the first one on the program that maybe has mentioned that as an experience. And not only that, but the way you framed it is that you envisioned it as a good transition from academia to more of the pace that you would see in industry. And so, and you keep alluding to this, well, it was much higher pace than what we were used to what you observed at Cornell, right? And the work that you were doing there. And so did that pace prepare you for the industry job that you eventually got? Did it help you succeed because now you had to learn how to do everything in a ramped up way than you had before?
I think so. I think the key thing for me was that in that kind of environment at the NIH, we work with a lot of clinicians, which you could do in a large academic center that might be if your lab's in a medical school, for example. But there was quite a bit of that interaction with clinicians, a lot of translational work, as I recall. And that really was important for getting into industry. That's really as a life sciences PhD, you learn the science, you do research, you could pick up new techniques, teach yourself anything in any field. How do you interact with others have quite a different background, whether it's coming out of business or, might be on the clinical side or legal, IT, you name it. And so that was really, I think, one of the ways that it prepared me for a career in industry, because, I sit on the science side of it. And how do you communicate that to others who are your peers who may not necessarily have the same technical background as you? But I often found even, at the NIH, very, very intelligent, talented people that, they understood everything that you would say. And that's been my experience throughout, even in small biotechs and now at a very large organization, that you work with a lot of people who have a different background, but, are very, very smart people. And they, totally get the science and everything else. So,
And Heinz, do you recall in your NIH days, do you recall having mentors, and do you recall any particular lessons that you learned either from them or with them during that time?
I think a lot of that mentorship, I mean, the short answer is yes. The advisor of the lab and some of the other staff scientists learned a lot from them. But a lot of the mentorship that really sticks out in my mind has been from when I've been in industry, and, having some really good former bosses who taught me a lot. Kind of how to play the game, so to speak, with, different things. I've always been, since my postdoc in highly regulated environments. So you have to overlay what you're doing in the lab to comply with what are really guidelines. And that may be something that's kind of, maybe a little nebulous or confusing to new PhD graduates, that you get this vision that the FDA or some other regulatory agency has a rule book. And they tell you exactly what to do. Like, you culture the cells this way and add, 50 microliters of this buffer. And it's not like that at all. It just says, come up with a system to culture cells, and that's it. And so that's where I really, a lot of the mentorship kind of helped me really learn, like, how do you do your job within a quality system. And so that is where what kind of sticks in my mind over the years is helping me to get where I am now.
That's awesome, and that's so amazing, because I hope everybody captured what you just said, which was pretty neat in terms of, like, the discovery process of your role and what you're supposed to be doing, and not only the discovery process of the science, but to understand what position you play and how you're supposed to play it with others, and this whole world of regulatory. um, Take us to that time and remind me again. So you were in Bethesda and you decided, well, I think there is an opportunity in Boston because of a startup company or a smaller company that had a position, and you just said, okay, let's just go do that. Or what? Take us back to that time.
Sure. Yeah, kind of, Tommy. So after my postdoc, I did work for a few biotechs in the DC area.
Right.
And so one of those companies was my real first exposure to what's known as good laboratory practices, which is a formal regulation. Sometimes people use that term GLP colloquially when they really mean best practices, but there is a formal GLP that applies to preclinical work. And the move to Boston was actually my wife had a job opportunity there. And so we went from Maryland to Boston for that. But we knew that, given our career paths in life sciences, that, no better place to be, that there was plenty of opportunity. So we made the move up there, and that is when I got involved with more like human clinical diagnostics, developing the assays.
Yes.
And so there too, you have another regulatory landscape that would be is what's known as design control, or you hear people say Part 820, which is just citing the section of the code, massive code of federal regulations that governs the design of the assays and how you validate them and all that sort of thing. And so that's really, that knowledge has really helped me in my current role, where I'm kind of on the other side of it now, where we run these tests for clinical trials.
Yes.
So it's good to have an understanding of how they're made, how they're validated, what the chemistries can do, and then how the constraints that the manufacturers are on. And then, of course, in clinical trials, now you have another good practice known as good clinical practice, or GCP. And for a lot of, for newer people coming out of your PhD, if you ever see a job description that says GXP, what they're really asking for is any kind of experience in GLP or GCP, or if you're in a company that manufactures something, it would be GMP for good manufacturing process. And a lot of these regulatory standards, it's enough to make your head spin. But they're all very common in what they're trying to do, which is essentially get you to document every step you've taken, so that if somebody wants to come in after the fact and in hindsight, they could retrace everything that you've done. And, just like you might make some conclusions in your testing and say, okay, I met this requirement, like as a good example is, you're developing an assay and you say the limit of detection is, X copies per mil. Somebody could review your data and either say, okay, yes, you demonstrated that, or I'm looking at your results and no, I disagree.
Right,
And
right.
so that's a lot of what you learn in these kinds of environments, is how to kind of think like that, where you're tracking every step. And really, like, what that means for your day to day is you're just coming up with a process. And so it's not all that different from being in an academic lab, where you'll figure out how to do experiments, and you'll develop an assay, and just for yourself, you'll come up with a process, and you'll keep a lab notebook so that somebody could, in theory, trace everything you've done, and then you'll put it into a publication. And it's not all that different, in terms of thinking of it that way than what you would do. It's but a lot more paperwork.
But so I think maybe that's what scares people. They think that, oh, okay, I'm going to go from the university where I have my lab book and I have lab meetings and this and that, to GMP, GLP environment where I'm going to need to be documenting what type of underwear I'm wearing or whatever it might be, like, I think people think, oh, this is going to be a daunting task that is going to be minutia. Is a reality, Heinz, or is there training? Is it just part of the culture? This is just how things are done, and maybe you can shed some light into that and dispel some of the myths of what they really want you to do and don't want you to do.
Right. I mean, it will do a lot more documentation than you would if you're keeping a lab notebook in an academic environment. But if you would imagine, let's say you're developing a PCR in your academic work, the very first time you record everything in your lab notebook, you might be going into the detail of, the components of your master mix, how many microliters of the enzyme, the magnesium, you name it, for a 50 microliter reaction. And then the subsequent times you're recording that, you just say, see page 10. So imagine if you didn't have the ability to just say, see page 10 every time you set up a run. You had a form to fill out and you would document everything you've done. Does it slow you down? Yes, it absolutely does. But if you design a process so you think through all the steps of what you will do in an assay, whether you're running it as a service where you're testing some type of sample, or you're designing a product that you will sell, it's really just kind of thinking through your process and coming up with what the pertinent information is to capture. So in my little master mix example, you might record exactly what you're adding to the tube. You might have a form that lists all the samples you're testing. Then you'd have things like the serial number of the pipetter, the lot numbers of the reagents. So there is quite a lot to do. We generally don't capture the color of people's underwear, but some days it does feel like you're getting into that. But that is part of it, and that enables an auditor to come and retrace your steps so that it's fully transparent. And so if you're doing these types of tests for a patient where the results, could be life or death sometimes if you get it wrong, that's what you have to do. And most people who are in this kind of environment are always putting themselves into the patient's shoes, that if somebody were running this type of assay on you where the result was going to guide your treatment for cancer, you really want to make sure that they got that right. And so that's the reason, that type of regulation exists. And that's the reason why that companies in that field will, absolutely comply with it. And it's not something that's done grudgingly or, it's really the culture of any company in this space is that, we respect compliance and we're all for it. And, that's certainly the case where I am now and past places that I've been.
And that's so awesome to hear too, because if it's part of the culture, is you're just going to adapt to that culture, and it becomes what you do. And I think it's so neat to be able to trace back and know exactly what you did, when you did it, with what tools, with what techniques, how long you waited. And that's really the preparation, I think, that we instill in our students as they get their training is record, put it down, write it down, make sure other people know about it, make sure other people can see it, and so on. But Heinz, you mentioned something when you were describing a little bit about your journey, which I want to be able to touch on, and so that the audience understands also a little bit what it is you're talking about. First of all, let's go back to the in vitro diagnostics, the IVDs that you developed, and if we can, just briefly explain what it means to be working under a CLIA waived lab and a non-CLIA waived. What are the different scenarios? How does that work? And we all became, I guess, for lack of a better term, part of the clinical trial for nasopharyngeal PCR test. I hope everybody now that that the population of the world now knows what PCR means I'm sure Kerry Mullis would be proud of this moment if he were still alive. But take us back a little bit. I mean, what is the IVD? What is this in vitro diagnostics, and how does that relate to the CLIA certification and how you can operate them? If you can.
Sure, that's a great question, Tommy. So CLIA is an acronym for Clinical Laboratory Improvement Amendments. And that's a regulation that is mediated by Center for Medicare and Medicaid Services, or they just go by CMS. And there are kind of these non-governmental organizations that have what's called deemed status. So the College of American Pathologists is one, or and you'll hear people say CAP-CLIA a lot of times. So if you were to go get blood drawn to a Labcorp service center, the testing would be done in a lab that's CLIA. Or if you go to IU Health, the same thing. It's all these are labs that are regulated by that, and it's not so much the government that is inspecting. It's really this group CAP, which is sort of a peer review system for the various labs. And so those regulations govern the testing. So if you have a clinical lab, and let's say you wanted to create your own test for SARS-CoV-2, you would create what's known as a lab developed test, or LDT. And you would follow the guidances provided by CAP. Like, they literally give you a checklist and say, when you validate an assay, you have to do this, this, and that. And you would document it, keep records, and every year you would be inspected by peers who are coming from another CAP-CLIA lab and make sure you're doing what you're supposed to. So it's a system that works really well for clinical diagnostics. Now we look at it IVD. So an IVD company, these are your Roches, your Abbotts, others who are manufacturing a test kit. So they're going to send you a box of reagents that has everything you need to run it. They usually manufacture an instrument to run it as well. And a lot of these tests are now fully automated, where the end user just puts a tube onto the instrument and it does the rest for you. So certainly we've learned a lot about that through the pandemic, when you'd hear all these companies being mentioned in press briefings and everything like that. But that they are governed by a completely separate group of regulations, because they're not really testing patients. They're manufacturing a product that they're going to distribute all over the United States. And many of these companies will market these all over the world. So, other countries have similar regulations that govern the design and manufacture. So when you hear IVD, in vitro diagnostic, that is something that the FDA runs. And if you compare that to a lab developed test, that's something that CMS ultimately would oversee. And there is a little bit of controversy about whether the FDA should consider what a clinical lab develops as the term we would use as a device, your medical devices. So that's still a little controversial, so I won't go into that. But an IVD company would sell their product to a CAP-CLIA lab, and then the regulations cover, if you're going to buy an IVD, there's certain things you need to do before you start testing patients.
Technical difficulties.
Sorry, Heinz. Sorry, can you hear me now? Yeah, my audio ran out of battery. Oh, so that's so cool. That's really neat. And you mentioned it's peer review. So somebody from another CAP-CLIA lab comes and they make sure that you're doing what you're supposed to be doing. That's so interesting. I didn't know that that's how it worked.
Right. Part of the deal, like if you are accredited by CAP, is that you agree that you're going to abide by their guidances, but part of it is you also have to be willing to go inspect other labs. So it really, in addition to the kind of scary part of somebody inspecting you, there is a lot of collaboration. So labs learn from each other. There's, you know, a lot of dialogue that you get that way. Whereas when the FDA or a government regulation is reviewing, it's not so much. That's more really strictly about compliance, which sounds scary, but it really is not. You know, if you have a good process in place, then you're okay. But you know, that's kind of, you know, how those two different worlds are connected.
And so when you were in these biotech companies, you were doing the validation and the testing for these in vitro diagnostics in preparation for FDA approvals?
That's correct, Tommy. So during my time in the Boston area, that's a lot of what I did was we would do, in being in a startup, you get to wear many hats. So you start from the beginning to the end, and you learn the whole process, like really accelerated learning compared to if you were in like a much larger company, you would kind of be stuck, not stuck, but it's in one area. But we did everything from, it was all infectious disease work. So it was designing oligos, you know, optimizing the conditions, porting it to the instrument, and then doing the real regulated piece under design control, where you're doing, and first you analytically validate your assay, which is just looking at, you know, limit of detection or precision, or whether it would cross-react with other bugs or something like that. And then you would work with another CLIA lab. So the IVD company would go to a CLIA lab, usually a hospital lab or one of the national labs, and you'd say, you know, we're developing this new assay. We know you test for this bug. Can we work with you where you split the samples and you run your assay as standard of care, and then you give us part of it, and we just see how it works on our device. And you would have results from the standard of care. You would compare the sample with the gold standard. So infectious disease, usually the gold standard would be micro. So you try and culture the organism from a clinical sample, and if it's there, you'd say, okay, detected. And then you would compare it to your assay to see if it were detected, and so on. And so that's the kind of work you would do in an IVD company. And then a clinical lab would do similar kind of work for a lab developed test. But the CAP CLIA regulations are more operational. It refers to a lot of the qualifications of the technicians. So you have to have certain credit hours in math and science and experience in a clinical lab. There's also a lot about, HIPAA compliance with identity of patients and things of that nature, how you report the results of testing. So they're really kind of complementary. And the controversial part is always that LDTs are perceived. And I think this is only perception, that they get a little easier when it comes to validating, but that's not necessarily true. A lot of clinical labs do as much as a IVD manufacturer. The one difference is that if you create an LDT, you can only run it in your lab. You can box it up and sell it, and you are really your device manufacturer at that point, and the FDA will pay you a visit.
So that's really, I mean, my gosh, Heinz, it sounds like you really did wear a lot of hats at these companies. And the way you talk about this, you've lived this, you've breathed this, you have a lot of experience, and knowing also the minutia of how to go about it. Did you Did you get specific training, or did you just, as you were going along, that you had these mentors and people that taught you the different things and what, what to do, what not to do? And I mean, it's incredible your level of knowledge, even as to what is the level of personnel that is required to be within, accreditation or certification for that lab.
So all pretty much on the job training, Tommy. And, for those starting out in their career, if they're wondering, a startup is a great place to go. If you want, if like use IVDs as an example, if you really had an interest in that, going to a startup would be a great place where you'd learn all these things in the course of a few years. Usually, a lot of those, a small company that's a diagnostics company developing IVDs would probably have some, more senior leaders who've come in an industry for a while, and you would really just pick their brain constantly and be able to learn from that. And you kind of learn the ropes that way. And when you live it day in and day out, driving toward milestones and things like that, there's no other possibility. It just sticks with you. And it is kind of a lot of fun to create those assays and see the finished product. And definitely, like when you get the approval letter from a regulatory agency, it's almost like learning when you publish a paper and you finally satisfy all the reviewers. It's kind of like that.
Yeah.
So
but it's kind of like that, but I can also imagine that now you can really start making a broad impact, right?
Yeah.
Because that opens up your ability then to start selling the product to other labs and having them also run it in their labs as well. Is that right? With the proper training?
Correct. Yeah.
Because you also have to get the training part approved by the FDA, isn't that right? You don't, not only let's say, so if you're just doing a PCR, okay, well, a PCR is fairly routine. Everybody knows how to do that, right? But if it's something fairly new, you have to put instructions on how to run that. Let's say it's a I don't know, another in vitro diagnostic, that it's a urine type of diagnostic or something like that. They would need some training on how to handle the sample and so on and so forth, that would go along. So is that really, or is that not really necessary? Is that somebody else? I mean, or it is really all tied in together as that one product?
Right. It is tied in together. You mentioned it though. There's the instructions for use, or you'll hear people just refer to that as IFU, or it's called package insert. That is part of, like, you could think of that as part of the kit, and the wording, everything you put into that document, like it has to be, that is approved by the regulatory agency, and you have to control the version. So even if you were to update it, you can't just do that on your own and say, okay, this is Rev 2. There's a process for that. And that is what the end user, the CLIA lab, would use to train. And of course, companies that market these devices, they will provide their customers training on how to use the instrument and things like that, of course, and they provide tech support. And for the CLIA lab, it's very important to receive the package insert, and the rules are you have to follow what the manufacturer would tell you. And so if you could imagine that you have an assay that you're testing on plasma, let's say, and you go along and say, well, I'm going to use it for saliva, you're now deviating from the instructions, in which now you're using that test, so to speak, off label. You have to validate, now you have to do your own validation. So it gets a little tricky, but to your point, the instructions for use is provided by the manufacturer, and
Yeah.
the end user has to, has to be written in a certain style that is understandable.
Yeah.
And even, I think you mentioned this earlier about like CLIA waived
Yeah.
devices. So like for a lot of what you'd see in the drugstore, like these lateral flow type test kits, they also come with a package insert. And the idea is that the general, the layman would be able to read through it, and you'd have to write it in such a way that it is understandable to the average person who is not a scientist, that they
Right.
could correctly use the assay without hurting themselves or getting the wrong result or something.
That's exactly right. And it's the interpretation of the results too, where everybody is so afraid. I mean, that's where we saw a lot of the bottlenecks with SARS-CoV-2, is
Mm-hmm.
that you didn't want people just to be interpreting the results on their own, right? And
Right.
it's like, but also so interesting how there needs to be approval of that, in terms of for like, you got to have trials where people are just left in a room alone with the device, and they have to read the instructions and they have to follow it all and do it successfully and so on. I mean, it's so interesting how it is regulated, but it's done for the public safety. It's done
Yeah.
so that everything works, and it works like it's supposed to work, and people are not misusing it, or, as you say, using it off label, which happens all the time, right? And people misinterpret things because they are using it off label and so on. But it's so neat to see this whole process of developing these in vitro diagnostics. Now, you made the leap from the developing side now to this testing side at Labcorp. And so you went through the Covance to Labcorp transition. I'm sure that must have been exciting. I don't know if you're allowed to tell us a little bit about like, wow, like what happened here, and like two big companies coming together like this. What an incredible thing. I watched from the sidelines here, and my mouth dropped when I heard that news. And I knew there were a lot of other competitors out there that also, I'm sure they they got kind of taken by this news. But can you tell us a little bit, what does it feel like? What does it mean to go, you're working in a company, you're doing your job, and now you have this announcement that, oh, now we're another company, or we're going to be called another company because we've kind of, made a nice big deal and a partnership with this other company. So what is that like as you're working in industry, as you're working? Did you feel the impact at all of that, or was it just like, oh, okay? Was it a cultural shift?
Well, that's a great question, Tommy. So when I joined Covance was right at the time the announcement happened. So I've only ever known the company as being part of LabCorp.
Okay.
And from my observations, being in the company, the cultures were very similar. And, it really did not seem, granted, I was new to the business. So, at the time when the kind of acquisition happened, it seemed very mild to me. Like, it's not, people might get it, imagine like there's all these horror stories of, you're in a company and all of a sudden it gets purchased and, fire everybody. And, people might be thinking about what's happening at Twitter right now. It was nothing like that.
Yes.
And, I will say that, if you go into industry and you're in a startup company, a lot of times the goal is for that company to be purchased. And that's not a bad thing if you're an employee there. You could, really end up, if there was equity or something like that, you can end up doing pretty well. And so, usually when a small company is purchased, the larger company buying it, they oftentimes do want to retain the people who develop the products because you're the world's experts. And so if you ever find yourself in that situation, it's not necessarily something to worry about. Like, so, those types of things can go bad, or you may find that the culture has changed, but that's all part of being in industry. There is no change is the only constant, as people might say, that you have to be prepared that things will change over time. The market changes. There's mergers, acquisitions. Sometimes you might be in a business unit that gets spun out, and you are now, wait, I was part of this big company, now we're small. And it's just the way it goes. And it's not something you could really control usually. And you just kind of adapt to it. That's really the name of the game. You see how things go, and
that's awesome. And so how has your adaptation to Labcorp gone, meaning from development to now testing? What was the mind shift that you needed to do there?
Right. Well, one thing that prepared me, like I had been in contract development roles where we're developing things for someone. So you have that kind of sense of like customer, like you're doing this for a customer. That was sort of similar. Learning the operation was, there was a learning curve there. It's clinical trials, very unique kind of business. And the knowledge that I had from developing IVDs has really come in handy in this environment.
Oh, good.
We're now the end users of a lot of those tests. And so knowing how they work, how they're developed, that kind of thing is really has really been helpful. And that's some advice I'd give to to newly minted PhDs who are thinking about where they're going to go. That a lot of times in your career, if you think about you're doing one thing and what's adjacent to it, like who's your customer? Could you go at some future point go work for a company that's the customer of the one you just left? Something like that. You could certainly do, and that will help you. And a lot of people do take that kind of where you're going into an adjacent area in the work stream, so to speak. So that is always very good. It enriches you. It keeps things interesting.
And I can imagine you're a wonderful resource because your clients, you can talk your clients' language and you can be like, hey, we need to adjust this. You might want to look at that. You might want to take this or that as we're trying these out. But by the time you get them, they're pretty much ready for clinical trials, right? Am I understanding correctly there? Or are they still doing preclinical type of, validation? This is pre-FDA approval that you're doing, or this would be for, broader dissemination?
So in a device trial, if you're the CLIA lab running the device, where you're just testing the performance of the assay, at that point, the assay's ready for approval, like it's a prototype, or it's being manufactured.
Right.
For what we do in Labcorp drug development, it's pharma trials. Sure. And so we will, and the pharma companies are at the same stage, like they're doing these phase 1, 2, 3 for at least the area I'm in. But they would also do some preclinical work and development work before then. Some of that they would do in-house. Some of it they would outsource. Sometimes is a little bit of both. But generally, the phased clinical trials are outsourced, where you have patients being seen, samples being taken, and then that's where we come in as we run them. And so that is in a CLIA environment because you're the results of those tests are being provided to somebody's physician. And so and also a drug company trying to make sense of whether or not you're seeing the effect you expect of the drug, or if there's some kind of harmful event that you would have to know about right away. So it's that's sort of how we apply all those lab tests now that we run.
Heinz, so interesting. And so it's awesome how your career in really like knowing a craft well, and I'll just say that PCR is a craft, is, you know, you're lucky if you get out of the first try. But it's, you know, it is a craft that it seems that you've honed in and you've perfected, or that at least you know this technique so well that you're able to obviously develop new tools, new techniques around it. You said something though at the beginning that I want us to maybe go back a second, because I think there are people that would be watching this interview that might be seated in the same position that you were, and they might not know it. And this is that you knew you wanted to go into industry.
Yeah.
And I, if you can, was there something that you knew about industry that attracted you to industry, or something that you knew about yourself? What was it that you knew that you wanted to go into industry versus staying in academia?
Right. I was always drawn to the applied aspects of biology. In fact, my undergrad major was biotechnology. So, that kind of, that field of study almost lends itself to, commercial applications. And when I was an undergrad, I did do some summer internships at some pharma companies. I grew up in New Jersey, so there's a lot of pharma there to choose from. And,
Sure.
that kind of gave me a little bit of a taste of what is it like in these companies. And, I really enjoyed, those summers working in those environments. So that's where I kind of knew I liked science. I enjoyed research, and I knew, okay, I do want to go on to grad school, but I do not want to be a professor writing grants and that kind of thing. I want to go, apply all this stuff that I've learned to, human health. And so for for folks who are starting a PhD program, or maybe you're at the end, these days, you don't really have to do a postdoc. It was, when I came out, it was a little less, not as easy to make that transition. Like, even, industry jobs were kind of looking for stock, but that's changed from at least my experience. It can hurt you to do one. So if you're still on the fence, it doesn't hurt to take a couple more years, maybe move around, if you want to move to a different institution, that can only benefit you. But, really, if you're interested in industry, when you come out of a PhD program, networking with people who are in companies is a good idea. Doing these kinds of informational interviews with people, and one of the things to kind of pay attention to in your graduate work that you could kind of use as evidence that you'd be a good fit for a role is you hit on one about the documentation. So keeping a notebook, like if you were to apply for a job in a regulated environment, you're right out of school, they'd say, well, what experience do you have, working in a regulated environment? You could say, obviously you say, well, I haven't been in one, but I know that when I keep a lab notebook, I understand how that's kind of similar to what you have to do. And so to be able to make that connection, you do have to really research a bit about what a company does, and things like, if you use GLP as an example, like what is GLP? And so these days that information is at your fingertips, that if you're coming out of grad school now, you could very easily access this information. And through social media like LinkedIn, for example, reach out to people, or even search articles that are posted on LinkedIn where you could learn about all of this, just so you have that kind of knowledge when you're starting out. And so that could help you get a job. I would say the more important thing is to really help you decide if that's the way you want to go. And I would also say that if you're not quite sure, don't sweat it. You never know how your career will take twists and turns, or where you'll end up. And early in your career, it does pay to be adventurous and take risks before you have a lot of responsibility in your personal life. So going to a startup, if you're worried about, well, it's unstable, it might close down, do that early, or if you want to do that even mid-career, that's fine too. But I'd encourage people to kind of just really have an open mind when you research these things, and have an open mind about an opportunity. Don't overthink it.
I think that's great advice. And it comes probably from a lot of experience, which I think I can tell that you've lived it, and you've done a great job at succeeding through every process, Heinz. So congratulations, really.
Thanks, Tommy.
And really, global director. Just last minute here, tell us a little bit about what is this purview of the global director of laboratory operations? I mean, does that mean that really global, you're talking to different labs around the world and you're telling them how to operate, what primers to get, what's happening?
Sure, that's a great question. So most clinical trials, like all the big pharma companies, are essentially global. The trials will happen with patients all over the world. And so a company like ours has a presence, like in the industry lingo, you'd say our footprint is around the world. So we have labs in Europe and Asia. And so if you could imagine, like if you're going to McDonald's and you stop on the New York State Thruway and get a cheeseburger, and then you drive, and by the time you hit Ohio, you stop again at McDonald's and you get a cheeseburger, you'll notice that they taste exactly the same. The stores are exactly the same. And when you do a clinical trial, it's kind of like that same thing. If you're testing a sample from an arm of the trial in the United States, in Indy, when you do the same thing in Europe or Asia, you could rest assured that the test is exactly the same. And so that's where the global aspect comes from, where I oversee the operation, not in the detail of telling what primers to order. We have our guys know they could run circles around me with that, but they is really to make sure that everybody is kind of on the same page with how we run the operation. And that's there's a lot of CROs in this industry. They operate the same way, and the pharma companies who do the trials and research are also the same way. And that's kind of what it means. So I do have a lot of early morning calls with Europe, and sometimes at night talking to Asia. But one of the best things about it is to get to know people from different cultures and backgrounds, and you spend most of the time just on the phone or on WebEx or something, but you really come to know them very well, like they're your friend, from down the street. So that's great. And occasionally they do let me out to travel, but not in the last few years.
Well, I certainly hope you do get to do some more traveling here in your next few years. And it sounds like such an exciting job and such an exciting role you play. Thank you so much for all you're doing and for all the great work that you guys are doing there. Heinz Reiske, Global Director, Laboratory Operations of Genomics, Labcorp Drug Development. Thank you so much for taking time to tell us about your life, to tell us about your successful career so far, and wishing you a lot more success to come.
Thank you, Tommy. I really enjoyed doing it. And, your viewers could always find me on LinkedIn if they have any questions.
Absolutely. Well, thank you. Thank you again. And well, all the best.
Thanks, Tommy.
Thank you.